So hello students, with the continuation of our last lecture, that is the third week, we'll study in this part the allantois formation, the mesodermal fate, and the trophoblastic changes. And along with it, we'll do the summary of the gastrulation, that is the formation of the definitive endoderm and the notochord formation. So this is a small video for you. I'll mute it first. So in this, the formation of endoderm has been depicted. Over here, this is our bilaminar germ disc, the second week, at the end of the second week. This one is the precaudal plate, which has established the cranial and the caudal axis of the embryo. This one is the amnion surrounded by epiblast cell. This one is the hypoblast cell. This one is the Huesner's membrane surrounding the primary yolk sac. So we have amniotic cavity and the primary yolk sac over here, or the second on the primary yolk sac. Then after that, what happens is when we view it from above, this one is the amniotic cavity. So again, this is the cranial and the caudal end. This one is the flat embryonic disc. Now what happens? There is a formation of a primitive streak from the caudal end of this region of the embryo. There is a colosicle, there is a thickening of the epiblast cell, which or the extraembryonic cells, which will induce the formation of the primitive streak in the epiblastic cell of the bilaminar germ disc. So this one is the primitive node or the Henson's node. This one is the primitive groove, right? So this one is the cranial end. This one is the caudal end. So these will be the future oropharyngeal membrane. This will be the future cloacal membrane, right? So then what happens at this section, we will see the cut section at this level at the caudal, the cranial end of the primitive streak. So what happens? The epiblast cells, they will start delaminating with each other by the down regulation of E-cadherin, and they will be first incorporate into the underlying hypoblast cells. So now they will form definitive endoderm. So this is how the definitive endoderm is formed. Now, how is the formation of intraembryonic mesoderm? More and more cells from the primitive streak, they will delaminate and they will accommodate between the epiblast and the endoderm. So now it will form a mesoderm. So this is how a trilaminar germ disc has been formed. The outermost layer epiblast will organize itself into the ectoderm. So now there will be expansion of the mesoderm. Initially it was just between the two, then it will expand to cover whole of the embryo. It will cover the amniotic cavity as well outside the epiblast, and it will cover the yolk sac cavity as well. Now, how the notochord was forming? This is a brief video about how the notochord was formed. So this is now the trilaminar germ disc has been formed. This is the primitive pit. This one is the cut section at this stage. Now the sum of the cells of the mesoderm which are entering earlier, they will form a notochordal process. This is the primitive pit which is extending inside the substance of this notochord as a notochordal canal, right? Now this notochordal canal has divided this notochordal process into an upper and a lower part. This lower part will incorporate into this underlying definitive endoderm, right? So now there will be formation of a notochordal plate. Now gradually what happens? So over here you can see this will disappear. The lower portion has disappeared. Now there is a formation of neuroenteric canal. So it will provide nutrition to the rapidly differentiating endoderm as well as ectoderm. So now here is the notochordal plate formation. This has incorporated with the underlying endoderm, it will again roll upon itself. Here is the neuroentry canal. This is notochordal plate. It will again roll upon itself, hold upon itself, and it will get detached from the underlying endoderm. Now it will form a solid notochordal process. So finally, a notochord has been formed in the cranial region. Now this primitive node is acting as a spearman organizer as it promotes three germ layer formation. And this notochord is found as a remnant in adults in the form of nucleus pulposus and apical ligament of dens. And the ectoderm overlying this notochordal process will be induced to form the neuroectoderm or the neural plate. Now there how allantois is formed. This is also shown. There is outpouching of the yolk sac into the connecting stalk. So this caudal part will form the allantois. Now there are certain anomalies. First one is the holoprosencephaly. So at the beginning of the third week of development, when gastrulation is initiated, it is a highly sensitive stage for the teratogenic insult. Why? Because all the three layers are being formed, and since it is the third week, which corresponds to the around fifth week of the from the last menstrual cycle, so women she may be unaware of the pregnancy. So if at that time if she consumes large amount of alcohol, it may kill the anterior midline of the germ disc cells. So it may produce a deficiency of craniofacial structures as seen in this picture. So thus this anomaly is known as holoprosencephaly. So various organs system are damaged. For example, eyes are not formed, brain is damaged, and so the condition is known as holoprosencephaly. Then there are certain tumors which are associated with gastrulation, that is sacrococcygeal teratoma. So if the primitive streak it failed to regress by 25th day, it may persist in the sacrococcygeal region. So this consists of clusters of pluripotent cells. So it may give rise to a tumor which is known as sacrococcygeal teratomas, and these are the most common tumors of the newborn infants. Then is the formation of allantois. So what is allantois? You get a short note on allantois. It is an outpouching of the yolk sac into the connecting stalk. So how it is on the day 16, how it is developed? It is the yolk sac will simply outpouch into the connecting stalk. So on development of the bladder, it forms urachus. So finally, after the development, after the birth, this urachus will be converting into the median umbilical ligament. So what is the significance of the urachus? In birds and reptiles, it forms a reservoir of urine. In human, blood vessels of allantois will form the blood vessels of the umbilical cord. So clinically, we have three conditions. One is the urachal cyst, urachal sinus, and urachal fistula. So if the lining is only remnant and it is closed on both the sides, and it might be filled with blood or some fluid sometimes. So the condition is known as urachal cyst. If this lining tract is communicating either outside or through the bladder, one end is blind, then it is known as urachal sinus. And if both the ends, if the passage is persistent in the adult form, so it is communicating umbilicus to the urinary bladder, then this condition is known as urachal fistula. Now coming to the fate of the mesoderm. So if you remember, this is the notochordal process which is inducing the overlying epiblastic cell to form neural plate. Now the three germ layers has been formed. So this one is the ectoderm overlying the amniotic cavity or surrounding the amniotic cavity. This one is the mesoderm, and this one is the definitive endoderm surrounding the yolk sac. Now gradually, I told you the mesoderm, it will spread hole over the embryo, one which will be surrounding the epiblast cells and another will be surrounding the endodermal cells. So the mesoderm is arranging itself into three parts around the notochords. So notochord is mainly found in the head and the trunk region. Then the mesoderm arrange itself into the three layers, that is the paraaxial mesoderm flanking the notochord. So paraaxial mesoderm is found only in the head and the trunk region. Then is the intermediate mesoderm found only in the trunk region, and the lateral plate mesoderm also found only in the trunk region. So the mesoderm gets organized itself into three parts: the paraaxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. So the paraaxial mesoderm, it differs in head and in trunk. The mesoderm that begins ingressing through the middle of part of the primitive streak gives rise to the paraaxial mesoderm that immediately flanks the notochord. So this one is the paraaxial mesoderm. It will form the trunk region, and also the bands of cells of the head and neck region. These bands of cells will form the somites, and these somites are the block-like condensations of the mesoderm. So mainly the paraaxial mesoderm is going to give rise to your head mesenchyme, which will be supplemented by the neural crest cells, the cervical region, and mainly the somites. Somites will be forming the building blocks of a trunk musculature as well as the vertebral column, the body of the vertebral column. So somites form an about 20th day at the head and trunk border. The remainder form in the cranial caudal progression at a rate of about 3 or 4 per day, finishing on about day 30th. So एक दिन में 3 से 4 सोमाइट्स बनते हैं, 30th डे में इनका फॉर्मेशन रुक जाता है. Totally, there are 42 to 44 pairs of somites flanking the notochord, and generally just cranial region में बनते जाते हैं, and जैसे जैसे caudally बनते हैं cranial वाले regress होते जाते हैं. Finally, only 37 pair of somites will be formed. The intermediate and lateral plate mesoderm form only in trunk. So mesoderm lying immediately to each of the somite also segments and forms a small cylindrical condensation, which is known as intermediate mesoderm. The intermediate mesoderm will give rise to your urinary system and the genital system. So genitourinary system is derived from intermediate mesoderm, and paraaxial mesoderm gave rise to somites and the head and neck musculature. So, and finally, the lateral plate mesoderm lies lateral to the intermediate mesoderm. It remains unsegmented and it forms a flattened sheet that is known as lateral plate mesoderm. So, on 17th day, the lateral plate splits into two: a ventral layer associated with endoderm and a dorsal layer associated with the ectoderm. So the ventral layer, which is associated with the endoderm, is known as splanchnic mesoderm. So it will form the mesothelial covering of all the visceral organs. And the layer adjacent to the ectoderm will give rise to inner lining of the body wall and to parts of the limbs. So hence it is known as somatic or somatopleuric mesoderm. Because the splanchnic mesoderm and adjacent endoderm act together, they will form splanchnopleur. There is a condition which is known as cyrinomylia. So gastrulation, it may be disrupted by genetic abnormalities and toxic insults. So in caudal dysgenesis, which is also known as cyrinomylia, there is insufficient mesoderm formation in the caudalmost region of the embryo. So since the mesoderm of the caudal region contributes to the urogenital system and also to the lateral plate mesoderm, so the formation of lower limbs are hampered, the urogenital system is hampered, and lumbosacral vertebrae may also be abnormal. So affected individuals, they will exhibit hypoplasia or fusion of the lower limbs, vertebral anomalies, and renal agenesis, along with imperforate anus and other anomalies of the genital organs. So such condition is known as cyrinomylia. So what is the importance of somites in the mesoderm? Somites give rise to the most of the axial skeleton, including the vertebral column and the part of the occipital bone of the skull, to the voluntary musculature of the neck, body wall and limbs, and to the dermis of the neck. So thus the formation and segmentation of somites are of major importance in organization of the body structure. So over here in this figure, you can see this one is the forming neural tube, and this one is the somites which are being formed on both the sides of the neural tube. These will form the vertebral body, and they will enclose this neural tube which will form the spinal cord. This is the caudal part of the neural tube. Excuse me. Now is the formation of chorionic villi. So the trophoblast, it is characterized by primary villi, which was found in the second week, that is outer syncytial trophoblast and inner core of cytotrophoblast. Then when it was invaded by the mesoderm, extraembryonic mesoderm, so now it will form the secondary villus, which was found found in the second week. Which will be found in the uh, third week. So this newly formed structure is known as secondary villus. Now by the end of the third week, the mesodermal cells in the core of the villus, they will start differentiating into the blood cells. So small blood capillary system will be formed. So now this is known as a tertiary villus. So outer syncytial trophoblast, inner cytotrophoblast, the inner core of capillary system will form tertiary villus. Now there is another formation of anchoring villi and establishment of fetoplacental circulation. Now since our embryo is growing faster, so now amniotic cavity is the yolk sac fluid cannot alone meet the requirement of the growing embryo. So we need to have a fetoplacental circulation. So what happens? The cytotrophoblastic cell from the inner core, it will start invading the endometrium uh, progressively. So it will the cytotrophoblastic cells in the villi, they will penetrate progressively into the overlying syncytium and they will reach the maternal endometrium. So this one is the cytotrophoblastic shell. Now here they will establish contact with the similar extensions of the neighboring villus stems and they will form a thin outer cytotrophoblastic shell. This shell gradually surrounds the trophoblast entirely and attaches the chorionic sac firmly to the maternal endometrial tissue. So villi that extend from the chorionic plate to the decidua bacillus. Decidua bacillus is the part of the maternal endometrium where the placenta will be formed, where the embryo is attached. So villi that extend from the chorionic plate to the decidua bacillus, the part of the endometrium where the placenta will form, are called the stem villi or the anchoring villi. Excuse me. Now, what are the terminal villi? The branches which are given from the sides of these stem villi. So there are certain branches which are given from the sides of these stem villi through which the exchange of nutrients and other factors will occur. So these are known as tertiary villi. These are the tertiary villi. You can see the extensions from the sides of these anchoring villi. So these are the tertiary villi. The chorionic cavity in the meanwhile has become larger, and by 19th or 20th day, the embryo is attached to the trophoblastic shell by a narrow connecting stalk. Now, the connecting stalk later develops into the umbilical cord, which forms the connection between the placenta and the future embryo. That's all in this session. And in the third part, we'll be reading about the neurulation. So that will complete our topic. That is the third week.